I'm Betsy Green, Executive Director of the Georgia Youth Science and Technology Centers. I am so happy to introduce you to our organization and the wonderful work we do. For 31 years, GYSTC has been the leading nonprofit advocate in Georgia for excellence in science, technology, engineering, and math education at the elementary level. We advocate for our hardworking Georgia teachers with ongoing professional development training so they can maintain the highest skill levels to prepare our children for success. We advocate continuously for quality science education for Georgia children and their families with year-round activities for kindergarten through 8th grade students. Our reach is statewide with special focus on those children and communities who are underserved and need us most. We are enthusiastic and we are creative. GYSTC programs are available so entire communities can come together around science-based exploration, education, and fun. Today, I'd like to share one such program we are most proud of, Georgia Discover Camp. As you will see, it's a learning camp with a science twist. Any Georgia student can participate and every Georgia student will enjoy what we have in store. Please take a look.
Hello, my name is Will Dodd, and I'm the Regional Coordinator at Oconee River GYSTC just outside of Athens, Georgia. Now, I hope you, ladies and gentlemen, had fun yesterday with Mr. Aubrey Crook as you studied sound engineering. That was way cooler than what I had when I was a kid. I think I had a cassette tape with a 4-track recorder that I would play around with. But what you did yesterday was way cooler than that. Today, what we're going to do is we're going to discover how to make a microscope out of materials that you can easily obtain around your home or at stores nearby. Now, we're going to visit Sandy Creek Nature Center, which is also located just outside of Athens, Georgia, to do that. But before we get into actually building our microscope, I want to run through the materials you will need to have on hand to build the microscope along with me. So the first item you'll need is some sort of smartphone with a camera. You will also need a laser pointer, which are pretty inexpensive. I think I got two of these for about $7. We'll need the lens out of the inside of this specifically, so we'll get to that in a little bit. You'll need a hairpin and tape, and if you don't have those, you can substitute that for some good old-fashioned Play-Doh. So these items will actually help us build our main microscope that we're going to use today. Now, when we want to view items, I highly suggest just having some items on hand. I grabbed a couple things from outside. I just have some rocks and maybe a piece of wood, one of these sweet gumballs, and I have this really cool leaf that I'm excited to look at, which has something on it called galls, which I'll talk about that a little bit later. So just some items to view. Now, when we want to view smaller items using our microscope, such as plant and animal cells, we're going to need to steady our microscope. And to do that, we'll need to build a stand for it. Now, because we're just using things that we can find at our house, I'll show you the stand or the materials that I use to build my stand. I have some varying sized books, and I have this random quinoa package right here, which will serve as parts of my stand. To have a real microscope that we can actually see cells with, we are going to need to have a light source. So for my light source, I'm going to use a flashlight and just a small rectangle of white paper that I'm going to place on top of the light source just to dim the light just a little bit. Now, we will also need to make slides. And usually, if you're at school, you have slides that are made out of glass and then a little plastic slip that goes on top of the slide. We don't have that luxury because we're just using some things we can find at our house. So you can use any type of clear plastic packaging. So I have a strawberry package right here that I'm going to cut some pieces out of to make my slides. Now, I also need scissors, of course, if I'm going to do any cutting. And for plant cells, what I'm going to use to view some plant cells is I'm going to use an onion. Now, an onion is a great way to view plant cells because the onion cells are a little bit larger. They're about 400 micrometers. And by micrometers, what I mean is in each millimeter, there are 1,000 micrometers. So these cells are going to be about 400 micrometers. So almost half a millimeter is the size of these. So you can see those pretty well. Now, I will want to dye my cells. So in order to do that, I will need either some red or blue food coloring. Those colors seem to work best. I'll need a little medicine dropper and just some sort of cup to actually mix up the right ratio between water and my food dye. I'll also want to use some Q-tips, and the Q-tips are going to be used later because I want to view some animal cells, and it's pretty easy to obtain animal cells since I have animal cells in my own body. And what I can use is the Q-tips and swab the inside of my cheek to get some cheek cells, and we can view those with our microscope. Now finally, I want to view some microorganisms that are in pond water. So I retrieve some pond water, and the rule with pond water is the greener the better. You'll find a lot more life if you have a lot of algae growing in your pond water. So that basically covers the materials that we will be using to build our microscope. Now before we get into actually building our microscope, I want to talk just a little while about the science that we are going to use to view these small objects. Now specifically, I want to talk just a little bit about visible light. Now, visible light is almost in the middle of something that we call the electromagnetic spectrum. And the electromagnetic spectrum is a spectrum of radiation that is at different lengths of waves or wavelengths. So we have really long waves at one side of the electromagnetic spectrum and really short waves at the high end of the electromagnetic spectrum. So this spectrum, it goes from the low end being radio waves, next is microwaves, then infrared waves, then what we'll be talking about today, which is visible light. Now above visible light, we start getting into the territory where the waves are incredibly short, very high energy waves that can actually damage our bodies. So when we get up past visible light, we get into ultraviolet radiation, X-ray, and then gamma ray. So you may recognize some of those terms. We'll start down at the low end and just kind of review what these are used in. So radio waves may make sense because radio waves are received by the radio in your car. So you can actually tune in to different frequencies to get music or stations on different areas of that bandwidth. Now, something fun is the sun is actually transmitting radio waves at Earth all the time. And if you tune to one of those in-between stations and you hear that sound, that fuzz sound, a lot of that is actually radio signals coming from the sun. So if you wanted to have a listen to the sun, tune to one of those in-between radio stations, and that's what the radio waves from the sun would sound like. Next, you get into microwaves, and that may sound familiar because a lot of you have microwave ovens. Now, microwave ovens, they transmit microwaves at the food that's inside of it, which vibrates the water molecules to heat up the food as it transfers that kinetic energy through the water molecules. Also, your cell phones, they actually transmit and receive in the microwave wavelengths as well. Now, above microwave, we get into infrared. Now, infrared is used at your home in your television remote. So if you look at the front of your television remote, you may see a little clear bulb that's in the front of it. That's actually what is used to transmit infrared waves at your TV to send messages to change the channel or turn the volume up. Now, after that's our visible light, but we'll get back to that in a moment. Let's skip ahead to ultraviolet. Now, ultraviolet radiation may be familiar because you might have heard of UVA, UVB, UVC listed maybe on sunscreen, the sun lotion that you put on yourself before you go to the beach or go outside. That's a good thing to do, is use that lotion because it protects your skin from that ultraviolet radiation. Now, ultraviolet radiation, the wavelengths are so close together that they can actually penetrate into your skin and sometimes damage the DNA that's in your skin cells, which later on can possibly lead to skin cancer. So that's why we even bother with putting on the sunscreen, just to protect our skin from the UV radiation or ultraviolet radiation. Now, up above ultraviolet radiation is where we get into X-ray radiation, which of course can be used to photograph bones or skeletal structure. Maybe you've had an X-ray before. I know I have, and that was fun to look at. And then finally, way at the highest end of the electromagnetic spectrum is gamma rays, which is terribly damaging to the human body. You definitely don't want to mess with those. But let's talk about visual light. So visual light, it does lay almost in the middle of the spectrum. And visual light goes from 400 terahertz to 789 terahertz. And just to make sense out of that, hertz are basically the measure of waves that pass a certain point every single second. So if we had something that was 1 hertz, it would just be one really, really long wave that passed a certain point in 1 second. Now, if we increase that to terahertz, that's going to be a lot. So 400 terahertz is actually the equivalent of about 400 trillion waves passing a single point. So they're very small waves. Now, that visible light spectrum, it goes, like I said, from 400 terahertz to about 789 terahertz. And you may have seen this visible light spectrum like every day when you open your eyes, but you may have seen the colors that are present in this visible light spectrum. If you've ever shined light through a prism and saw the rainbow that is refracted from that white light, you also might have seen this if you've seen a rainbow, which is, of course, sunlight shining through water droplets in Earth's atmosphere to act much like a prism in order to refract all those colors that are in the white light. Now, do you, ladies and gentlemen, know what colors are actually visible in the visible light? Now, you may be saying Roy G. Biv, which is an acronym for red, orange, yellow, green, blue, and violet. So of course, those are the colors in the rainbow, and each one of those has a different wavelength. That red would be down on the 400 terahertz level, and then the wavelength gets smaller and smaller up to violet, which would be around the 789 terahertz level. So something cool about visible light is, of course, it travels at the speed of light, right? Because it is light. So light can actually travel through space at 300,000 kilometers per second. Now, in space, there's nothing to slow it down. It's a vacuum. So if light actually hits something else, such as the gas that's in our atmosphere, it does slow down. If we actually pass light through water, it slows down even more substantially. Slows down from 300,000 kilometers per second to about 225,000 kilometers per second. And we can actually view this slowdown in real time if we take a look at a glass of water. So I've got a glass of water right here, and if I place a pencil in this glass of water, you can observe this phenomena. As you see, the pencil that's underneath the water appears different than the pencil that's above the water. And the reason for that is because the light that's traveling through that water actually slows down and is bent or refracted, making it appear larger. If you look closely at that pencil that's underneath the water does appear to be larger. And also, if you compare the pencil underneath the water, or how it looks under the water, compared to above the water, it also looks sort of like disjointed, like the pencil's almost broken. And that is really just as an effect of the light being bent or refracted as it travels through that water. Now, another item that we can bend or refract visible light with is a good old-fashioned magnifying glass. Now, these lenses, because of their shape and because of the speed that light actually travels through them, they bend the light or refract it and actually spread the light out so that the image on the other side of the magnifying glass appears to be larger. So if I look there, my eye might get really big there. So that's what's going on with these magnifying glasses. Now, today, we're going to use a lens that we're going to get out of a laser. We're going to use this lens, attach it to a smartphone, and make a microscope out of it. And in order to show you how to actually build your microscope, I want to take you to a place right outside of Athens, Georgia, known as Sandy Creek Nature Center. So let's head there, and I will show you how to build your microscope. My name is Will Dodd, and I'm the Regional Coordinator with Oconee River GYSTC. And today we're going to discover how to build and use your own microscope. Today's STEM discovery activity brings us to Sandy Creek Nature Center in Athens, Georgia. Sandy Creek is home to 225 acres of woodlands and wetlands, with the North Oconee River creating its western border. The Education and Visitor Center includes many live animal exhibits, many interactive history exhibits, and a resource library, all housed in an eco-friendly building. It may be difficult to obtain a quality microscope to view items up close, but today I will show you how to build your own using simple items you can find pretty easily, such as an old cell phone, an inexpensive laser pointer, and a hairpin. We're going to take a walk around the Nature Center to collect some items to view with the microscope that we build. And we'll start right here with this pond water. So this pond water is perfect for collection because anything that has this much algae growing in it is going to be teeming with microbial life. Now that we've collected a few items, let's head back to the classroom and take a look at them with the microscope we built. So now we're back in the classroom. We've got what we want to view with our microscope, and I'm going to show you what we're going to need to actually build our microscope. So first, I've got an assortment of old cell phones here, smartphones to be specific with cameras. I found that the low-end Android I have doesn't give me quite the picture quality that I can get out of an old iPhone 5. And the autofocus seems to work a lot better on this one too, so I prefer the iPhone. You're going to need, in addition to the smartphones, a PET laser. So I found these for pretty inexpensive, $7 for the two of them off of Amazon. You can also get this model for about $3 at the supermarket. But this one was far more easy to get the lens out of. You'll also need some Play-Doh. You need a hairpin, tape, a flathead screwdriver, and that will give you your actual microscope itself. Now for objects to view, in order to get plant cells, the best plant to start with is a red onion. We're going to use a clear plastic strawberry box to create slides for our do-it-yourself microscope. You're going to need scissors, a medicine dropper, some assorted containers, and some water. To get the animal cells that we're going to view, I'm going to use some Q-tips. Also, to view our animal and plant cells better, I'm going to use a little bit of red food coloring, as well as a flashlight with a just small sheet of white paper, or small piece of white paper. Also, to view, if you guys remember from the trail, we picked up a couple items. I got a sweet gum ball here. I've got a clover flower and an actual clover. We have a little piece of a fern, a dandelion, and I've got a lichen right here that we're going to check out as well. I also picked up that pond water. We're going to have a look at it as well. And of course, you might want to get some paper towels as well. That's always a good thing to have on hand. So now that we have all our parts and everything you need, we're going to go ahead and build our microscopes. So first, we're going to need to disassemble the laser in order to get the lens out of it. So I'm going to unscrew the top of the laser. And then I'll find this metal piece that I can unscrew with the flathead screwdriver. And once that piece is removed, I should be able to flip the laser itself over and get a spring and a lens, which is what we're after, out of the laser itself. So I'm going to go ahead and set these pieces to the side. And with the lens, I'm going to take my hairpin and pinch the lens into the hairpin as shown here. Next, I'm going to want to take a look at the laser lens itself, and you're going to notice that one of the sides of the lens is more rounded than the other. You're going to want to face the rounded side of the lens toward
your cell phone camera and perfectly line up that lens with the camera. Now at this point, I want to tape my hairpin to the phone itself. Next, I can go ahead and power on the phone and access the camera to actually see the microscope image. Now at this point, I can just carry around the microscope and view different items that I would like to see. This is the first way to attach the laser lens to your smartphone. Let me show you a second way. So for this method, we'll need the laser lens itself, smartphone camera, of course, and Play-Doh. Now we're gonna get just a small piece of Play-Doh and roll it into a thin line, or a Play-Doh snake. Again, I'm going to want to look at my laser lens and find the side of the lens that is more curved or convex and line that side up with my cell phone camera. Now at this point, now that I have my laser lens lined up with my camera, I can take this Play-Doh and actually wrap it around the laser lens itself and press it against the back of the cell phone so that the lens stays in place. So at the end of that process, I should have my laser lens stuck onto my cell phone camera using the Play-Doh.
Now, just in case that building part went a little too fast for you, I just want to let you know, some of this might get a little complex. So what I'll do is each phase of the building, I'm going to repeat again, because that we're live, you don't really have the advantage of stopping and rewinding the video. Now, even though I'm going to repeat the build twice, if you still get lost at this point, keep in mind that this video will be uploaded to our website at a later date, to which you can go back and view this as many times as you need just to get your microscope working. Because I found that a lot of trial and error does tend to go along in the building process as you're trying to get your microscope set up correctly. So quickly, I'm just going to run through the build of the basic microscope again, just so that you have the chance to build that along with me and use it in this video. So first, what I'm going to do is get the lens out of my laser like I showed you earlier. And to do that, all I have to do is just unscrew the top of the laser itself. Be very careful not to turn it on. You don't want to shine it in your eyes or anything. I'm going to then take the flathead screwdriver and unscrew the slotted piece that covers the lens. And once I have that taken off, I can simply dump out the lens itself into my hand. Now I can continue on with my build by taking my cell phone, and just like I told you in the video, if you look closely at the lens itself, you'll notice that one side is sort of curved and the other side is flat. Now we want that curved side facing the lens of our camera. So what I'll do is I want to attach this lens to my hairpin first, and then I can line up that curved side with my cell phone camera. So what I'm going to do is spread my hairpin and place my lens inside the hairpin just like that. Now I'm going to look at my lens and make sure that that curved side is lined up perfectly with my smartphone's camera. At this point, I simply grab some tape and tape the hairpin in place where it's at. So then all I do is turn on my phone's camera app and I'm ready to view some items. Now, just in case you don't have the correct type of hairpin that really pinches on well to your laser lens, what you can do is use just a small bit of Play-Doh to do the same thing. I can lay my laser lens on top of my phone's camera, as I showed you earlier, and simply get a small piece of Play-Doh, roll it out, sort of like a little Play-Doh snake, and then wrap it around the laser lens just to secure it to the actual smartphone. So with this, we've got our basic microscopes that we can use to view any number of objects. So let's head back over to Sandy Creek Nature Center and take a look at some of the objects that I collected there.
So a really easy item to look at with your microscope is just the back of your hand. So let's take a look at what I see. All right, so to focus my microscope, it's pretty simple. Basically, I move the camera lens closer and farther from the object itself, and that will focus my microscope. So let's take a look at some of the plants that we picked up at Sandy Creek. First, I'm gonna grab a clover flower and zoom in to see what we can see, what details we can see on the clover flower. So a clover flower is a composite flower, so if I look closely at each one of these small white parts of the clover flower, I'm actually looking at an individual flower, and I can see its petals. And I can actually see down inside that tiny flower. Or if you want to, you can look at the stem itself and look at the tissues that actually bring nutrients to that flower. Now I'll look at the sweet gum ball we picked up. Let's take a look at our fern. So with this close of magnification, I can actually see clearly the veins in the leaves of the fern. Let's take a look at this lichen. Lichen is actually a symbiotic relationship between two different organisms. It is a fungus, but inside of it, there is actually algae that's able to live outside of an aquatic environment. I can actually see these hair-like structures coming off the lichen itself. Now, something you see every day, we can take a look at a dollar bill and actually see some of the tiny features that we may never notice on the dollar bill itself. I know when I was a kid, we always looked at the number 1 in the top right-hand corner to see if we could find the spider. Now, from far away, it definitely looks like a spider, but close up, not so much, just a funny pattern.
I want to take just a little bit more time to view a couple other objects. So I have a rock here, just a rock sample. I'm going to see if I can show you a few of the things I can see on my rock sample. So first, again, all I did was turn on the camera app on my smartphone. And to focus my smartphone microscope, I can just put my lens as close as I can to my rock sample. And maybe you can see what I'm seeing. Now, at this magnification, I can actually see the individual small pieces of the minerals that make up the rock itself. Now, some of these, I can see a little bit of light refraction off of them. I'm getting a little bit of a rainbow effect there. Now, if I wanted to zoom in even closer, I can use the camera's zoom feature and just drag across the screen, and I can get even more magnification and get even closer to the object that I'm looking at. So I can get a pretty good look at this rock just using that. Now you notice probably the most challenging part of viewing an item with your microscope is keeping it steady. So we'll get into that just a little bit later. Now I also have a piece of wood here. I think this is just some wood mulch that came off of a playground. But I can view that just by moving my microscope lens as close as I can to that wood. And I can actually see the minute wood grain present on that piece of wood there. It's pretty wild. Now, something that's really fun to look at, I was excited when I found this. This is, I believe, an elm leaf with something on the surface of it that are known as galls, and that's spelled G-A-L-L. Now, galls are pretty interesting because they can be, they can come from insects actually working with the leaf themselves, or from funguses that can grow inside the leaf itself. But these particular galls, if I'm not mistaken, I want to say are caused by maybe aphids or some other insect. But basically what happens is the insect can bite or even lay eggs inside the leaf itself, and they release chemicals that encourage the leaf to grow a structure around the organism and basically make a home for it inside of the leaf. And that's a gall. So I can take a look at these galls. A lot of times you'll find some that are caused by wasps, and they have the young of the wasp inside them. And I found a few before that actually had some larvae inside it. So let's see if I can focus. And again, to focus, I'm just moving my lens close to and then far away from my object. So I can get a really good close shot of that gall. And you can see the leaf is encouraged to grow almost like a woody structure, like a hard structure on its surface, which is really unusual for a leaf. But again, that comes from the chemicals that are released by whatever insect was working with this leaf. Now, I did crack one of the galls open, and it looks like these galls might have been abandoned a long time ago, because when I look close at it, it really just looks like a dark, empty, woody structure on this leaf. But yeah, if you can find some of these in your yard, they're definitely something worth checking out. So feel free to use your microscope to look at many different objects. One fun thing to look at with your microscope is even just your fingerprint, because you can see very, very close up the minute details in your skin itself. So definitely check that out. It's worth seeing with this microscope. Now, we just used our microscope to view items just by holding it close to the items themselves. We can view even smaller objects with our microscope, but in order to do that, number one, we need to be able to keep our microscope steady so it doesn't move around a lot. So in just a moment, I'll show you how to build a stand for your microscope, which is not going to be made out of complex materials, just some books and things that I find. I'm also going to show you how to make a light source for your microscope, because we will need to shine some light through our samples in order to see them better. All right, so we're going to head back to Sandy Creek Nature Center, and I'll show you how to build slides that we can use for our microscope, as well as a stand to keep our microscope steady.
So now we are going to use our microscope to look at the cells of an onion. So to do this, we are going to have to prepare a slide the best we can with materials we have at home. So the slide material itself, I made my slide just out of a plastic strawberry container. So any type of packaging that's a thin piece of clear plastic you can use to create your slide. So I just used scissors and cut out of this strawberry container a rectangular piece about this size, and then a smaller rectangular piece about this size. So we'll use those in just a moment. Next, we're going to need to use a flashlight to actually light the slide from underneath. But in order to use this flashlight, we are going to need to tape a white piece of paper over the top of the flashlight itself. So I'll go ahead and put a piece of tape on both sides of this piece of paper. All right, now we have our light source for our microscope. We're also going to need a stand for the microscope itself. So for my stand, what I found worked the best was 2 random books stacked on top of each other. And in order to stick the flashlight or the light source for my microscope onto this stand, I used some of the handy Play-Doh that I already had. All right, so I balled it up and was able to stick it on the stand just like that. I'm gonna go ahead and turn on that light. So I was able to just stick it in place right there. So next, I'm gonna need something to hold up my phone. Now, I found that this jar of quinoa was the perfect height to set my phone on and position my lens over that light source.
So again, just so you can build along with me, I'm going to repeat that process just so I don't leave you behind. To make our slide, again, I'm going to use just any plastic packaging, which again, this is a strawberry container. And all I need to do is cut out probably a 1" by 2" rectangle from the plastic. You can see there, I cut that out, and I have an edge on there, which I don't really want that edge. I'd rather it be flat. So I'm going to cut that edge off and discard it. So I have my large rectangle, and if you've ever used microscopes at school, a lot of times you'll have a rectangle of glass about this size, a glass slide about this size that we use, but again, we're going to use just clear plastic. Now I'm going to cut out a rectangle about 1/3 that size, so maybe about 1" by 1" or smaller. So I just have a small square of plastic that I cut out of my strawberry package. So that gives me my slides that I will use a little bit later. Now I need to steady my microscope so that I can view these small objects. And to do that, I'm just going to use a couple different books. I found that I had to troubleshoot a little bit, and you find different thickness books to make my light source stand up high enough to actually match the height of my microscope itself. So for my light source stand, I will use these two books. I'm going to create my light source like I showed you from a flashlight and a white piece of paper. And what the white piece of paper does is just dims the light so it's not as intense. So I'll bend that over just like I showed you earlier and tape that paper onto my flashlight. So again, that's just going to dim my flashlight so that when I turn it on, it's not as intense. And to attach my flashlight to my actual stand, I'm just going to use a ball of Play-Doh. And that will do a pretty good job of holding my flashlight in place. All right, so I'll just go ahead and set my flashlight inside of that. So there's my light source for my microscope. Next, I found that this quinoa package right here did a pretty good job of holding my microscope. So what I can do is move the package just close enough so that the lens, my laser lens, is centered over my light source. And the cool thing about the packaging I'm using with this screw top lid, I found that I could actually unscrew this lid and get just a little bit of height. So it kind of acted like a fine focus for my microscope on accident. So if you wanted to use something with a lid like that, you can go back and try that out. Now I will say, this does take a good bit of trial and error. You may have to go back and adjust the heights of different things just so that you can get your microscope at the correct level above your slide so that your picture on your screen is focused enough. Now, at this point, I want to show you how to prepare a slide with specifically the onion so that we can view it with our microscope. So at this point, we'll head back over to Sandy Creek Nature Center and I'll show you how to prepare your slide with the plant cells from an onion.
So next, I'm going to get a small slice of this onion. I definitely suggest getting an adult to help you with this part. Now that I have my slice of onion here, what I can do is pick out one individual slice or layer of the onion. And on the inside of that layer, there is a very thin membrane of skin that I will want to peel off. Just like that. Now I'll take that membrane of skin and take my larger piece of rectangular clear plastic, put that piece of onion skin on the slide itself. Now that we have the onion skin on the slide, we will need to dye the onion skin so that we can see the individual cells. Now, usually in a classroom, we'd use methylene blue to make this happen, but at home, you can just use red or blue food coloring to do the same thing. To do that, I want to mix 1 part food dye to 1 part water. The way that I'll do that is just put 2 drops of food coloring into this container. And then I'll drop 2 drops of water into the same container. And this is just a really simple way to mix up our dye into the right ratio. Next, I want to use my little medicine dropper to suck up some of the dye and put probably just 1, maybe 2 drops on my actual onion skin. Now, once I have that dye on my onion skin itself, I can take my smaller piece of clear plastic and gently press it over the onion skin itself. That will make my slide look like this. So some objects, such as our onion skin cells, are so small that we need to shine light through them in order to make them visible. The best way to do that is by flattening them out, as we did with this slide.
So as promised, just so I don't lose any of you guys in this complex process, I'm going to repeat that onion cell prep that I showed you just a moment ago. So I have my slice of onion here. And just as I showed you, on the inside of any of these layers, there's a thin membrane of cells that I can gently peel off to actually get a thin piece of onion cells. Now I got a really small piece, it's on the end of my thumb, and what I'm going to do is get one of my slides that I made and press the onion skin onto that slide so that it's very flat. And as mentioned, I will need to dye these onion cells so that I can actually view them in my microscope. So to do that, what I want to do is mix 1 part red dye or blue dye and 1 part water. So I will add 2 drops of red dye into this little container here, a little measuring cup. And then I'll add 2 drops of water. And you can definitely play with this ratio. If it ends up being too red or too blue, you can definitely add more water if you'd like to. But I'm going to stick to this 1-to-1 ratio, which is going to make it very red, but I should be able to see the outlines or the cell walls of these onion cells very, very well. So I'm just going to add 1 drop of this red dye to my onion cells here. And then add my top piece of my slide, as you see right here, and that's going to smush these onion cells very flat so that I can shine light through them. Now to view my onion cells, I'll want to place the slide on top of my light source. So I'm placing my slide on top of my paper. And then I'm going to line up my laser lens right on top of the slide itself so that I can view my onion cells. Now, it looks like it's not too bad out of focus right now, but you may find that you have to raise or lower the books that your light source is on just to have the image of your slide visible in your screen. So what I can see if I can take a photo of that and then show it to you guys. I can see vaguely my onion cells in this photo. Now I'm zoomed all the way out. So if I place my microscope back on top of its stand, I should be able to use my camera's zoom feature to zoom in even farther. So let me do that. I'm going to zoom in as close as I can. And at the closest zoom, I'm getting about 150 times magnification with what I have here. So if I can zoom in correctly, I'll try to snap a photo and see if I can show you what I see. And feel free, while I'm finding this image here, feel free to snap photos of what you see in your own microscope and share them with us on Facebook and on Twitter, and be sure to tag us so that we can see what you see. So here's an image right here. You might be able to see what I can see, but I can clearly see the cell walls of my onion cells, and I can even see some of the nuclei, or the nucleus that's inside of the plant cell itself, which is pretty cool for a homemade microscope to be able to get that down that far. Something else that we can view besides plant cells is we can view animal cells. And as I mentioned before, I have animal cells in my body, so I can retrieve animal cells pretty easily just by using a Q-tip. And what I can do with a Q-tip is swab it inside my cheek. So this will be real pretty. I'm just gonna swab it around like that, get a lot of cheek cells on there. And then I'll get another slide that I made, and I'm going to smear that saliva cheek cell mix all over this slide, in the middle preferably, so I know where it's at. So I just smeared saliva and cheek cells onto the slide, which I can't really see them very well, of course, because with the naked eye, they're way smaller than those onion cells that we just looked at. Onion cells are about 400 micrometers in length, which is about half a millimeter, less than half a millimeter. Cheek cells, human cheek cells, are far smaller than that, ringing in around 60 micrometers, so much, much smaller. So I'm also going to dye these cells as well with my red dye, and they're going to show up as very, very, very tiny, small, usually circular shaped objects under my microscope. So I've got my cheek cells. I put my red dye on top of my cheek cells, and then what I'm going to do is take my top slide and smush down that cheek cell and dye mix. Now, this one may be a little bit more challenging to find the actual cells than it was with the onion cells. So it may take just a bit of time, as I said, trial and error, just to locate your cheek cells. You may even have to do an additional swab just to try to get more cheek cells. But let's see if I can locate anything with what I found. And so far, just by looking at it, I can tell, let me snap a photo here. I can tell already that my dye is far too red. So I would have to definitely decrease that concentration of red in order to see my cheek cells. So with the amount of time we have, I think I can try one more time. So of course, any ideas on your part, what do you think we could do to actually decrease the concentration of the red dye? Pretty simple question, right? All we would do is just add more water. So I'm going to rub off this sample, and this is highly irregular lab procedure, right? Usually you would wash these off really well in a sink or something like that. I'm just rubbing it off. So of course, I'm definitely contaminating my sample of cheek cells, so we may get some odd stuff and smudges in there, but I'm just gonna try again for the sake of being able to see some cheek cells. So I will go ahead and add many more drops of water to my mix. Mix that up. I'm actually going to add some more. And I'm going to use this and see if we can get a better sample of cheek cells. So I will grab another Q-tip. And again, do the pretty part, right? I'm moving it around in my mouth. That's nice, right? And I'll again smear this Q-tip onto my slide. And once again, I'll go ahead and add a few drops of my dye. Smear that around a little bit. And maybe we'll get lucky on this one and see some cheek cells. All right. Maybe you're having better luck than me with the cheek cells. Hope you are. I'm going to open up my camera app again. And, oh, lost my slide. And, you know, that's normal with making your own microscope. You're going to drop a lot of things. So I'm going to move my lens. And I'm vaguely seeing tiny, tiny pink-colored circles that are my cheek cells. So I snapped a photo, and you may be able to barely see those tiny pink circles. Those are my cheek cells. So that's pretty cool. See if you can get your own cheek cells visible on your microscope that you made. Now, the most fun item to view with this microscope is by far pond water. So in order to show you some pond water, I want to take you back to Sandy Creek one more time just to show you what I was able to find in the pond water that I found there. So let's head on back to Sandy Creek.
So if you remember, back at Sandy Creek, we picked up some pond water, and we're going to put a few drops of this pond water on our slides. And the thing about pond water is there's a lot of life that we can't see, microscopic, but we can see it with the microscope that we made. So I'm going to put a few drops on the slide, like I said, flatten it out so these little organisms don't have a whole lot of room to swim around in, and see if we can capture some images of them on our microscope. So I find the more debris you can get into the slide itself, the higher the chance you're going to actually see some action on these pond water slides. There we go. I see some movement. I do believe that's one of our paramecium, which is a single-celled organism that gets around by wiggling these little hairs called cilia. And they get around pretty quick, so you have to look close so that you don't miss them.
All right, like I mentioned, pond water is definitely the most fun item to view with our microscope. It's a lot like fishing because you get one sample, you look around all over the slide, you might find one organism, you might find no organisms. And then you'll get another sample and you'll find maybe 10 plus organisms in one slide. So depending on the pond water, definitely, you're going to get more or less organisms. But keep at it because you're going to get some really cool pictures and some really cool videos from this. So I've got my pond water, and as I said, the greener the better because you're going to find more life in this green algae-filled water. And I am going to get a couple drops. Well, I don't want to get red in there. I'm going to get a couple drops of my pond water. And I'm going to try to get it as close to like the debris-filled algae as I can. And I'm going to place maybe one drop, 2 drops. I'm going to go with a couple drops there on my slide. And again, I'm going to place that slide cover on top. And what that does is it smushes out that water, makes it a thin area so that those microorganisms don't have a whole lot of room to wiggle around in. It makes it easier for us to be able to shine light into that area where they're at. So I'm going to center my lens over my microscope slide again. And I'm going to zoom in and see if I can see anything moving around. Now on the video, you all saw me, I spotted a little paramecium, but at the end of the video, I spotted something called a macrothrix. That last organism that you saw moving around on the camera was called a macrothrix, and it's a crustacean. So crabs are crustaceans, lobsters are crustaceans. That's a microscopic crustacean that has a shell and it moves around in pond water, and they get around pretty quick. If you can find one, they're a lot of fun to observe swimming all around. So as I said, this is a lot like fishing, and I am not seeing any microorganisms in this sample. So the good news is we've got a whole bottle. So what I can do is really the proper way to clean this off would be to go take it to a sink, rinse it off, dry it really well. But since we're live on camera, and this is my only slide that I have left, I'm just going to smear it off on this piece of paper, so probably not the best way to do it. And go ahead and see if I can get another sample. And like I said, you want to get as much debris, as much gunk into your sample as you can. So I got a little bit of algae stuck to this one. So let's see if I'll get lucky with this. And I might not, because like I said, it's like fishing. Sometimes you get something, sometimes you don't. Now, one of my favorite things I was ever able to catch and see with my homemade microscope was a mosquito larva. Now, if you're familiar with mosquitoes, they have an interesting life cycle. They start as larvae that are aquatic. They live in water, and they're tiny, tiny organisms that swim around and feed in the water. But once they pupate, I think is the term, they turn into a pupa. They actually emerge from that pupa as adults, the adult mosquitoes that we know and you kind of hate. I'm not a big fan of them myself. Carry diseases a lot, right? They're a lot of fun. If you can get one of those larvae underneath your microscope, they are action-packed. They wiggle all over the place and they're really fun to watch. So see if you can catch one of those if you find any mosquito larvae around your home. And you probably want to get rid of the mosquito larvae around your home since they do carry diseases. So let's see, I've got a piece of debris. I'm going to zoom in on it. And I'm not getting a whole lot in this sample. Let's see, let me zoom in a little bit more. I can see the algae very close up. Let's see if I can snap a picture of the algae. It's moving around, so there might be something in there. I'm just moving along. I'm moving my phone and moving it closer and farther away, just kind of tilting it up and down. And I got a great snap of this algae. You may be able to see it. It's just very thin green threads. So I can see that very well with my microscope. But I'm going to just try maybe for 30 more seconds to move across my slide. And as I mentioned, if I ever so slightly tilt back and forth my camera as I'm holding it on the stand here, I can sort of focus in and out of what I'm looking at. So it's just very slight tilting motions are going to give me focus as I move my smartphone up and down. So I'm not seeing anything moving around a whole lot in here. Actually, nothing at all. But I could keep at it. So at this point, I hope that you, ladies and gentlemen, get to look at many, many different objects with your microscope. I hope that you get to spend many hours even looking at pond water, because you'll definitely find a lot of cool organisms in that pond water if you keep at it. And one of my favorite places online, there is a website by Nikon, and you can find an actual catalog or gallery of microscopic pond life videos. And these videos, you can click on them online and actually see the different microscopic life moving around so that you can more easily identify what you're looking at. So definitely see if you can find that. See if you can find some microorganisms in your pond life. And I hope you had a whole lot of fun making your own microscope. And if you did, I highly encourage you to look into some careers that use microscopes on a daily basis. So some of those careers include things like chemists, biochemists, zoologists who study animals, biologists who study all life, and probably one of the most fun that I've seen is forensic scientists that actually use their microscopes to solve crimes. So definitely check out those careers. I hope this activity encouraged you to be more interested in STEM careers. And I hope that you'll join us tomorrow to learn more about the principles of flight with Dr. Bob Young. We're going to build a lot of different paper airplanes. So please join us tomorrow. Again, my name is Will Dodd. I'm the regional coordinator at Oconee River GYSTC. And any pictures or videos you take on your microscope, please share them on Facebook or Twitter and tag us at GYSTC. So see you tomorrow. Bye.